Hi, it's Ms. Parrott, and this video is the second in a series of 3 videos about the Central Dogma of Biology. That was described in the first video. This second video is going to focus on how messenger RNA is translated so that a protein is actually formed. Of course, when that protein does its job or doesn't, a phenotype will be expressed. The basic idea of translation is that when messenger RNA arrives at the ribosome, depending on what the mRNA code is, different amino acids will be brought to the ribosome by transfer RNA. That polypeptide will grow, and then when it has finished, it will release and do its job. The ribosome is made up of 2 subunits, a large one and a small one, and the messenger RNA is going to flow through it. These molecules are another type of RNA called transfer RNA. And transfer RNAs are the ones that bring those amino acids, this is threonine, histidine, and methionine, it will bring those to the ribosome from the cytoplasm. Let's look a little bit more deeply at this principle. First, we need to recognize that messenger RNA is broken up into sections of 3 RNA bases called codons. So of this 15 base long section, there are 1, 2, 3, 4, 5 codons. And each codon corresponds to a specific amino acid. For example, here I have methionine. Methionine can be notated by just the letter M, and AUG is the codon that corresponds with methionine. So this is chemically what methionine looks like. Then you have glycine, right? Letter G, its codon, GGG, and that's its amino acid structure. And then finally, aspartic acid, whose the letter code for it is D, and the codon that codes for aspartic acid is GAU, right? Guanine, adenine, uracil. And then here is the structure of aspartic acid. So depending on the codon, a different amino acid will be brought. Now the amino acids, of course, are brought to the ribosome, like we said, by transfer RNA. And here is a little bit on the structure. As we know, RNA is a single-stranded molecule, but this doesn't look very single-stranded to me. But actually, it is. It's just that it's folded in on itself, and so there's complementary base pairing going on within the tRNA molecule. At the bottom of each tRNA is a section called the anticodon, and it's called that because the bases here are the opposite of what the messenger RNA codon is. And so we need this anticodon because when the tRNA comes to the ribosome, it needs to be able to attach in and match up with the corresponding codon. So transfer RNA is going to bring our little amino acids, depending on the codons, right? AUG, CAC, ACA. So right now we have ACA as a codon, and this codes for threonine, so our little tRNA is dropping off the threonine here. One thing important to note is that all polypeptides, all of them, begin with the sequence AUG, which is for methionine. There you go. And so this polypeptide is going to grow and grow. These amino acids are going to continue to join until you reach a special kind of codon called a stop codon. And there are actually 3, UGA, UAG, and UAA. So whenever the mRNA sequence gets to one of those 3, the polypeptide addition stops. The polypeptide flows into the cytoplasm or into the endoplasmic reticulum to be further folded. And those are the principles of translation.